Hi, everyone, and welcome. I'm very excited today to bring forth some of the data that both Dr. Kim and I have been working on to develop animal models for DESSH. And with that, I'm going to share my screen, a PowerPoint that put together with our data in it, and kind of talk through it here. Just to give a few introductions, Dr. C.H. Kim and I are both working on trying to develop these models, one in mouse and one in zebrafish, and each one of these animals will have its own advantages that we can try to use. I'm based in Grand Rapids, Michigan at Michigan State University, and my lab has been working on developing the mouse model. And Dr. Kim is based in South Korea, and his group has been working on the zebrafish model. And so far, we have been able to successfully make these. I'm going to show you some images today. And the big questions that we're trying to answer here, and what I'll show you some of the data for today, are understanding if either one of these models or both of these models can recapitulate some of the symptoms associated with DESSH. And the things that we studied in our initial run were craniofacial behaviors relevant to ADHD and autism, as well as susceptibility, to seizures. And we've now started looking at the brain to measure common brain cell types and trying to understand if there's certain regions or developmental time points that we may need to look at in more detail to really get a good understanding. And to that end, we're trying to develop more strategies to look at the root causes and, again, these specific cell types that might be changed or altered in DESSH. I'll start with the mouse model. So here in panel A on the top, this is just a cartoon kind of showing where we were able to manipulate the mouse's genome. And we used a technology called Cre-LoxP. These are kind of like molecular scissors, where we were able to cut out part of the gene, creating a WAC heterozygous mouse. So we lost one copy or one of its alleles. The mice develop pretty well. You can see a picture of him down here in B. The heterozygous mouse is about the same size and looks pretty similar to the wild-type mouse overall. And in C, this is just a validation. So these bands are actually the pieces of the mice or the mouse's DNA. And the lower band where it says recombined allele, this tells us that we are actually cut out part of this DNA because that's smaller compared to the wild-type allele. So it's one way that we can look at the mice and kind of understand who's who as well at the genetic level. And we also took this DNA and got it sequenced just to validate the locus, and that's in D. This is just a DNA sequence of the allele. So we feel pretty good that the mouse recapitulates, at least at the genetic level, what a knockout or a heterozygous mouse should look like. And while we were doing this, Dr. Kim's lab is also developing zebrafish. So using a technique called CRISPR-Cas9, where it will give you a targeted deletion, but kind of a random mutation in there. And what they were able to generate a few mutants shown here in E. If you look at the red site, 2 of these actually had a deletion and where they targeted WAC, and this led to a truncation or loss of their allele as well. And in F and G, they were again able to make viable zebrafish that look pretty good. And G is like what I showed you earlier, their validation at the DNA level, that they can actually detect who is who between these two. And so one of the first things we wanted to understand is whether or not some of the features that have been described for DESSH could be recapitulated with these animals. And one of the most striking ones, I think, that we consistently found were there were alterations in the frontal bone in mouse and the jaw in the zebrafish. And this was interesting because both it starts very early at around the day they're born. So P here represents postnatal. That's the day that the mice are born. And what we notice is if you look in A and B, and then A prime and B prime, the green area represents the area that hasn't fused together yet with the 2 frontal bones. And it's a little wider in the WAC hets, and this persists into adulthood. So they actually have a larger frontal bone area. And we quantified this and in E through H, and these are just, each dot here represents a different animal, but as a group, I hope you appreciate that we do have a shift in the frontal bone width in the animals. And similarly, Dr. Kim's lab was also able to recapitulate this in the zebrafish, and that's quantified here in K. So the good thing is we do have one of the features consistently found in both of these models that we might be able to work with in the future. Another thing we looked at were various behaviors. There were several behaviors that were not changed. And that's a good sign because if everything was changed, I would worry more that maybe the mouse couldn't see or hear or something else was wrong, but they do perform pretty well. So did the zebrafish. Some of the things that did change in the mice were differences in social preference and differences in some short-term memory tests. And these are in A and B. Just to give you an idea here, one of the common things we do is we give a mouse a choice between a novel mouse and an object. And they usually always prefer the mouse over the object. So the red dots here represent how much time we spent. So the higher it is, the more time. And wild type mice, this line here just represents that there's a nice separation between these. But in the whack het, there's a slight gray area where mice are kind of shifted towards spending a little more time with the object, maybe a little less time with the mouse. It's very mild, but it is something that we could try to follow up on. And then we did a maze called the Y maze, which tests short-term memory and attention. And we do see some changes in this one as well. So we have two behaviors in the mice that could potentially be used to test in the future. I think a very strong one that the zebrafish shows is this color map in C and D here. When these fish are put in a novel tank as a group, they usually stay very close together. And this is what's called social cohesion. In the whack knockouts that Dr. Kim's group has made, you can see just by the heat map here in D that there's a lot more spread out as they're swimming around. And this is quantified in E. So there's a pretty strong social behavior in the fish that I think could be advantageous for us there. And they also move a little faster. They have a little bit of hyperactivity as well. And they noticed this is just measured here in F and G, just how much they moved within the tank itself. And then one of the last things we did to understand some of the core features are challenge the mice with a drug that we know will increase brain excitability. And we have a scale that goes from 0 to 8. As the higher the number, the more severe the seizure. And what we found is both in female and in male mice, this drug actually elicited more severe seizures compared to the wild type mice. And this was very consistent and very strong. We think that there is a good reason to use the mouse model, at least, to try to understand brain excitability. So this is something that we're looking at right now. And this prompted some other questions that Dr. Kim and I are now looking into. And one of the first ones is just looking at various targets in the brain that could be changed. One of the things I can say in both models is that the brain seems at the gross level very normal. And that's good because it means that all the cell types are there. There's nothing missing, nothing out of place. When we look at the brains, it's really hard to tell the difference between them. There were some subtle markers that we think are off a little. One of them is this protein called parvalbumin. There's slightly less in the whack hets. We don't think the cells are missing. It's just this is a protein that can be modulated by brain activity. So we're wondering if this is associated with the elevated susceptibility to seizures as well. And it also is a subpopulation of what's called GABAergic neurons. These are the inhibitory cells of the brain that kind of calm down excitatory events. And consistent, but a little differently, Dr. Kim's lab found that one of the GABAergic enzymes, GAD1B, is also slightly less expressed in the brain region of zebrafish that's similar to the brain region that we looked at called the forebrain. So we think we have some initial biomarkers that we can start looking at, and we're going to dig deeper into this as well. And then I'll just end with kind of some things we're trying to do and what we want to do in the future. And um, again, we want to figure out what brain cells are involved um, uh, and associate maybe if these are underlying things like seizures or behaviors or other things like that. We're now collaborating with some friends at UC Davis and trying to look at genomic techniques to understand if there are actual signatures or other biomarkers that we could pull out that these could possibly be used for future therapeutics. We're also trying to assay all the ways that these symptoms could arise and understand the causality. For example, I showed some GABAergic marker changes, and we also talked about seizures. And the idea is that if the GABAergic markers are less, and these are your inhibitory cells, that might cause some of the seizure susceptibility. So that's something we're also interested in. And honestly, if there's any other ideas, I hope today is a good chance for everyone to talk and meet. And I'm sorry I can't be there, but I look forward to meeting you all. But let us know. Reach out to Caitlin, reach out to anyone in the group, and feel free to contact me anytime. I appreciate your time and thank you.